So let's take a look at this anaerobic jar here. This is a jar for cultivating anaerobic bacteria on Petri plates. And it's actually exactly the model that we have in the microbiology lab, just that you don't get to use it anymore because we're doing everything online. But let's take a look at this thing. So it's basically a jar. It has a lid that's airtight. And in the lab, just picture this, you have your Petri plates, you get to streak out different types of anaerobic bacteria, and then you would put them upside down. Instead of putting them in the incubator, you put them first into this jar. Once everybody has loaded the plates that they want to have in this jar, then we would add a packet, and this is like this envelope. It looks just like what you see here on the slide. So it's an envelope that gets opened up and it releases CO2, and it also releases hydrogen gas. So the hydrogen gas then will combine with the available oxygen that's still in the jar. It will combine with oxygen to form water. And the CO2 is just basically also displacing sort of the existing environment that contains a lot of oxygen, what is adding the CO2, and the water, the hydrogen will combine with the oxygen that's in the jar and will remove the oxygen that way. As soon as the oxygen level drops to where anaerobes can actually grow, this indicator, we have a little indicator strip, always looks similar to what you see here. It's an indicator strip and it contains methylene blue. And methylene blue is a chemical that turns colorless when there's no oxygen present. So if it's reduced, in its reduced form, it has no color. On oxygen, on regular air, this strip would be blue because it contains the methylene blue, and methylene blue is blue when it's exposed to oxygen. So this is a good indicator for the environment in the jar. As soon as the methylene blue turns colorless, you know you're going to have an environment that is anaerobic, and then your anaerobic bacteria, they should start growing. The whole jar will be placed in the incubator, incubated at 37 degrees Celsius, and then we would come back 2 days later and take a look at what's growing in there. And you typically have to open the jar under a hood because these anaerobes, they create gases like you won't believe. It smells like socks that haven't been washed in 3 months, and it's not very pleasant. But anyway, so I will be putting a anaerobic growth lab online that you can then follow through and at least theoretically do some experiments. But the anaerobic jar would have been a big part of this week's lab for the microbiology lab. And that will be going along with inoculating bacteria in liquid medium, which contains, if you want to go back to this slide right here, we have the fluid thioglycolate medium. It's basically a broth, but it contains the thioglycolate and it removes the oxygen, and then you can grow your anaerobic bacteria. But anyway, so it will be all theoretical. Here is an anaerobic chamber. We don't have anything like that in our lab, but if you work a lot with anaerobes, then you would have to do an anaerobic chamber. It's very useful. Here's also, these are culture techniques for capnophiles. Capno refers to CO2. And if you have a microbe that requires very high CO2 concentrations, then you need to grow them similar to the anaerobes. So you just add more CO2 to the atmosphere of that jar. You can also use a candle jar because the candle will burn up all the oxygen that's in the jar and then release more CO2. So that's another way of doing it, but I'm not sure how many people are still doing it that way. Most people will add a CO2 packet. And here, sort of a sideline topic on the special culture techniques. There are sometimes biosafety levels attached where you have to then use extra precautions when you're growing certain microbes, and the levels go from 1 through 4. Just quickly, so biosafety level number 1 is basic teaching labs, like what we mostly do. Biosafety level 2 requires lab coat, gloves, eye protection. That's what we require in the microbiology lab. And biosafety level 3, you're going to have cabinets that will prevent airborne transmission, so mostly laminar flow hoods. And then biosafety level 4, that's where you're going to have negative pressure labs. So if anything would actually happen in that lab, the air will flow into the lab rather than out of it to release any kind of chemicals or viruses or whatever. So those are just for your information, biosafety levels. Yeah. Here are some people working in a biosafety level of type 4, of level 4 laboratory. That's the kind of precautions that you would use for working with coronavirus, for example. So you see people in these full body suits, gloves, everything sealed. That's biosafety level 4. Okay, let's move on to the selective and differential media. So, by definition, selective media, they suppress the growth of unwanted microbes and encourage the growth of the ones that you want, your desired microbes. So they contain inhibitors to suppress the growth of those microbes that you don't want. Basically, you're selecting a certain type or category of bacteria and allowing them to grow while you're suppressing the growth of the ones that you don't want. In contrast, differential media are those that differentiate between closely related types of bacteria by having them generate certain colored colonies or distinguishing them based on certain metabolic characteristics. So differential media will enable you to differentiate between closely related bacteria. For example, Enterobacteriaceae, they're very closely related, but you can create media that will make, let's say, an Enterobacter grow pink colonies and E. coli will grow shiny purple colonies. So let's take a look at this. Now here's an example. Here we have blood agar, and there you're growing bacteria, and depending on the type of hemolysis that they can do, so lysing of the red blood cell, you can differentiate between different types of hemolytic bacteria. So some will show beta hemolysis, others will show different other types of hemolysis. Here's also differential medium. There you are differentiating between two closely related bacterial species. That would be a Staph aureus here, which changes the pH of the medium, and now you see that what's growing here turned the agar medium yellow because of acid, and here the Staphylococcus epidermidis didn't do that. And so now you can differentiate between two closely related Staphylococci. Moving on to enrichment cultures, those are growth media that help fastidious organisms to grow. So those are the picky eaters. They want certain nutrients that are normally not present in general growth medium. And so you're encouraging the growth of a microbe that's typically a picky eater. So here would be a summary of the different types of culture media. Chemically defined, you know all the ingredients and their amounts. Complex media, that is good for most chemoheterotrophic organisms. Those were your beef broth or nutrient agar. Then we have reducing culture media, like the fluid thioglycolate medium. That's for obligate anaerobes. That's what they would, like an FTM. That's what we have in the lab to grow for it. to grow the anaerobes. Selective media, they suppress unwanted microbes, encourage the desired microbes to grow. Differential media allow you to differentiate between closely related species of bacteria. And enrichment media are for those picky eaters where bacteria require certain special ingredients in order to grow. So the Pure Culture Challenge, that is an experiment that you luckily got to finish in the lab. And that is a way of separating out cultures that are in a mixed culture. So what you want to do in the end is you want to have a pure culture that contains only one species or strain. Typically that's done with the streak plate method, so that you are diluting bacteria on a streak plate until you have isolated colonies. And a colony is defined as a population of cells arising from a single cell or spore or from a group of attached cells. And such a unit that can form a colony is fittingly called colony forming unit. Now, you were given a mixed culture, and you were supposed to hand me pure cultures, which all of you did and got to finish that. The streak plate method is the way to go, and that's what this looks like. That's what you actually did in the lab. You were streaking out the original culture, a loop full, and that's on section 1. Then you re-sterilized your loop. You went into section 1 and dragged some bacteria over into section 2, which is kind of spreading them over in section 2. And then you re-sterilized your loop and went back on your plate and took a little bit of section 2 and were dragging it over and spreading it out in section 3. And then this is pretty much what you guys got. We used the Serratia marcescens, that's red here. Then you also had white colonies from E. coli. And you had the purple bacteria, the Chromobacterium violaceum. So you did exactly this experiment, and once you had isolated colonies that are labeled right here, you can circle 1, you went ahead and picked them off your plates and then were streaking them out on agar slants, and that's exactly how a pure culture, how you get a pure culture of something. Finally, here on preserving bacterial cultures, if you want to do long-term storage of your pure bacterial cultures, then you can either deep freeze them somewhere between -50 to -95 degrees Celsius. Most labs have -70, -80 deep freezers. Another way of preserving for long-term storage is called lyophilization. It's freeze drying, where you're dehydrating the bacteria and you're freezing them, and then storing them in a vacuum a lot of times. For the next section, we're going to take a look at the growth of bacterial cultures, including how bacteria divide. So we're going to take a look at binary fission, and then we're going to take a look at the phases of microbial growth and describe the relationship to generation time. So let's start out by taking a look at bacterial cell division. If you've ever taken another class in biology and you were looking at mitosis, you will appreciate how much simpler cell division is when you don't have to deal with a nucleus. So with bacteria, all they have to do is duplicate the genetic material, and then they're ready to divide. Under ideal conditions, they can do this maybe as fast as 10-12 minutes. So in general, the cell division mechanism of bacteria is called binary fission. Sometimes you also see a mechanism called budding, where the resulting cell, the new one, is much smaller and buds off of the parent cell. But in general, bacteria divide by binary fission right here. The conidiospores and fragmentation, that's kind of rare. So binary fission is what we're going to focus on. So here it is, a very simple kind of mechanism. The cell will need to replicate its own DNA. So DNA needs to be replicated. Once you have 2 copies of the entire genome of this bacterial cell, it basically can start dividing. Bacteria, they start to put down cell wall material sort of down the middle right here. And as they're finishing up building the cell wall material, that's when they can divide and split into two separate entities. So it's very simple. The process, again, is called binary fission. And so here we have an image showing a bacillus that's ready to divide. So you can see it's elongated. It has started to put down cell wall material right here and right here. It's not quite done yet, but almost. It's ready to divide, definitely.